that kind of stick out. It's a weird little U-shaped indentation.
Huh. Little
So
smelling
in the habit
periscope.
of in the habit of naming rockfish after their features, green striped, split nose, yellow eye.
Seems like the best way to do it.
We just passed a pineapple benthic siphonophore.
You got a net there? No, just kidding. That is just sediment.
When Jess looks in the cloud, is all she sees is fishing gear now.
Pretty much, yeah. Sediment did look like
I see.
We did not.
Yeah. It's a good thing to be looking out for.
Get all those squid.
Are those squid? In,
yeah, Argus cam, lots of it. We get some rocks. Crinoid
So
Point.
these are some Sebastomus rockfish, the white spotted reds. And Southern California has many species of those.
Marley, you want to open the iris a bit there?
Do you guys just lump all those, Erica, in counts? Video counts?
We tend to in our AUV images, yes.
Okay.
They can be a little bit difficult. Some of them are down to, does it have scales on its chin? And how long are the rays on the first gill arch? And things like that that are kind of difficult to establish from imagery. But some of the analysts, like Tom and his group and some of the other labs down there, have their own sort of gestalt for telling them apart. That's a Penatula sea pen.
Tom just speaks to them, so that's not entirely fair.
I've also had Tom say, well, they're greenish yellow, not yellowish green. And like that's a point of clarification.
He seems very self-aware about the concept, though.
He'll be delighted to know we're talking about him.
He's probably listening.
It's all right. He's not on science chat, so he can't retort.
But he's been doing it for such a long time that you really do get sort of a feel for it, and you know at what depths which species are going to be found, you know where they hang out, you know whether it's soft bottom or on the rocks, kind of who the other species they hang out with are, and you can narrow down your guesses for sure.
Yeah.
Yeah, taxonomy is a total art.
So these big sea stars are Stylosterias foriae, and they're the fishing sea star. Seen a couple of Paragorgia.
Any idea about the nature of that name?
I'm guessing they
like?
actually, yeah, I'm guessing they prey on some of these smaller rockfish.
That would be an amazing predation to observe.
In Argus cam, there's a lot of squids.
Yeah.
Yeah, I was just about to say the Argus cam view is pretty awesome right now.
We got a swarm of anchovies in the morning, and a swarm of squid now.
I know, I love it. I
squid.
Squid predation right there. That was cool.
Come here.
So is that the fishing starfish?
Yes.
Cool. Yeah, that's not the way I'd want to go.
Yeah, you want giant squid.
Yeah, yeah, that'd be a good way to go. Don't want any of that Aristotle's land or anything.
Some more of the Antipathes, Christmas tree coral.
The urchins are just so dominant. It's
Yeah.
a while now. Well, soul. So is it the Dover sole you still get at these depths, or I don't know if you saw
You
that
will
one.
see that. I think that one was a slender,
Okay.
and
Brownie.
they have more a chevron, a pointy tail, and usually it has a white edge on it.
On the tail.
You will see, yes,
Cool.
and you will see Dover up here, but they'll be much smaller than what we've seen down deeper.
I see. We're all taking advantage of your knowledge, Erica.
Meredith's saying that the small sea pens are maybe stylatula, not pennatula. So
I was gonna say. Yeah. Couldn't believe it.
Meredith got to it first.
Just joking.
but then there's also the taller sea pens. Have you seen those, Meredith? There's short white ones and tall white ones.
So we're talking about the habitats, and up until recently, we really didn't have a good map of the area. We didn't know. We only had a very coarse understanding of what the bathymetry would look like, where the features were, where the deep sea canyons and pinnacles were. And now we're about 98% of the way towards a completed map with high resolution bathymetry, and once processed, eventually, Ben will have that backscatter for you as well for the region. That'll make dive planning incredibly easier as we select and stratify our sampling to make sure we cover right amounts of all the different habitats.
Yeah.
Once we have that data, the bathy and the slope, the rugosity, the backscatter, we'll turn that into a classified benthic habitat map. So we'll map out across the sanctuary what proportion of the sanctuary where and in what configuration are everything from kelp forests to rubbly reefs, rocky reefs, soft bottom sediments, and so forth. So that's the next step. But this first step, just getting the bathymetry, was an incredible accomplishment. The Nautilus has been part of that acquisition since I started here, trying to fill that gap.
Oh, that's a big sea pen.
The guess on that one is a shallow Holypterus. There's two different species of Holypterus, and we're finding that one occurs shallow and one occurs in deeper water.
Yeah, the naming conventions are very helpful. Shallow.
Well, there were two species identified on the coast, and I think they were separated latitudinally. And now that Meredith's actually doing genetics on them, she's finding it's more split by depth rather than latitude.
Yeah, but the genetics just overturn so much with all these cryptic species. It's like, really?
And half the time they split it further. No, these two can only be told apart by genetics.
Doesn't count to me. No. OTUs, ugh, no.
And how much longer we have in this transect?
Yeah, we have 80 meters left.
Roger.
Yeah. We'll be getting, yeah, it should be pretty consistent. We might get a little slopier.
Roger.
Right now it's a little.
Slopier, nice.
Yeah. That's the data I have right now. Sure, the backscatter exists. If we have the multi-beam, we have the backscatter somewhere, but.
Yeah, I think it just hasn't been processed yet. And as we pulled together the bathymetry, a lot of it's stuff we've acquired recently, and it's been really great and high resolution. But some of it's really old data, and the backscatter is it's just not that helpful.
Yeah.
There's ways to collect backscatter to increase the likelihood that you can utilize it then for habitat mapping, and there's other ways to make it infinitely more difficult to work with, and some of that old data is just not great.
Yeah, it's all just a proxy still. It's nice to get down here and make sure.
Yeah, excellent point. Exactly.
So isn't there a large mapping project currently going on worldwide to try to fill in some of those gaps? Because I know only a very small percentage of the whole ocean's been mapped.
Yeah, I mean, on a first order, we just have satellite altimetry, and that is really coarse for kind of understanding the features that would be meaningful ecologically and otherwise. And then I know there were some, earlier in this season, we were given some priority mapping targets that, like this deformation ridge on the Cascadia margin was a priority. These kind of interesting areas that did not have the multi-beam. So yeah, to me, whenever I'm helping out with Nautilus mapping stuff, I feel like I'm making a global contribution, because no matter what, it's given to a global marine repository like GMRT, and we're aggregating from other ships' data, and we're path planning around what other ships and us have done. So to me, all this mapping seems like a global effort and a kind of not proprietary contribution that we're all gaining benefit from. So that's kind of part of the fun of it, filling in the gaps, and the rest is just moving forward. But yeah, I think it's interesting to me because, I think that's the statistic that people pull from when people say, oh, 80% or something of the ocean hasn't been explored. And that's still just the idea of having this bathymetric observation. And still, it's like the midwater, the most large largest habitat on Earth like that, that's not an interface that's so easily observed like that. Yeah, mapping's a team effort. I think we're actually going to switch out for dinner relief, but we have 50 meters left in the transect, so they'll finish that off.
Raj.
Just one more thought on that, Ben. That was a really interesting point. And, you know, if you think about it, one thing that we're not getting, right, we're just trying to get that first picture ever of the bottom of the ocean complete, right? But how much does that change? If you looked at the land 10 years ago, 5 years ago, 100 years ago, how much has it changed? How much has the Sahara expanded or the rainforests been cut down? Well, we have no idea how dynamic and changing the ocean environment is, and that's the bulk of the Earth's surface. So that first picture ever is revealing and so critical and so important, but we're so far behind our terrestrial counterparts in terms of understanding what's going on in the ocean, because that will just be one snapshot in time.
Right, and even then in more dynamic areas, it needs to be done more often.
That's exactly right. We know that there's rivers of sand out there moving to and fro, covering habitats and exposing other habitats, changing the distribution and diversity of creatures we see everywhere. That's exactly right. And we're just, in terms of technology, we have so far to still go, I think, in terms of looking at all those things.
We had colleagues that put a bunch of instrumentation out at axial seamount, and they came back in a year to pick it up, and it was all under lava flow.
Whoa. There's no retrieving that.
No, they
You must be
did
talking.
not get some of it back.
You must be talking about the rumbleometer that got buried.
Yeah, I think they lost a couple things.
We did recover that years later. We were poking around and came across it.
Axial?
Yeah. It's a picture of the recovery on the Ropos website. I think it's entitled Bringing Home the Bacon. And they got the data out of it.
Oh, really?
Years later, yeah. It was half buried in a lava flow.
Really? That's awesome.
That is amazing.
Nice glass bottle there.
Yeah.
You've got a ID on that one, Erica? Actually, I don't. That actually, yeah.
Red,
what
red
was
stripe,
it?
maybe?
Red stripe, maybe.
It looked like red stripe. So as we near the end of this transect, do you want to continue on to that intermediate waypoint just beyond Z1? Or will we be stopping to look at anything here?
I don't think I have any instructions that says we need a collection per se or a core, but I will wait for a second. If I don't see anything back in the science chat, then we will continue on,
okay.
As you mentioned, to that point.
Sure. Bridge nav.
Another green stripe, rockfish.
Let's do another step 100 meters bearing 353. Thank you. Just another 5 meters left to go in our transect.
Can you tell, have we done any cores yet?
We have done 2 cores.
Sorry, I keep forgetting I'm on mute. Yeah, we have 2 cars.
Great, thank you.
Okay, that's the end of the transect.
Copy, end of transect.
And still going 353.
Getting attacked by squid.
Yeah, squid rain.
So yeah, I don't think we'll do any course here. We'll just keep heading along to that intermediate waypoint.
Sure, sounds good.
Let's wait for Argus there.
Roger.
Or you can tilt up a little, maybe. It's better.
What was the little rockfish? Did you see that little rockfish associated with the urchin? Kind of had stripes, vertical stripes, Erica.
I think I'm seeing
No.
split nose. I'm sorry,
Still
short
split
belly.
nose.
Would you
Short
like,
bellies.
would you like to look closer? Or?
Not right now, but
There's another one.
oh yeah,
Right?
to the right,
Maybe.
actually,
Yeah.
yeah,
Yeah.
to
Can
that, to the
zoom
right.
in there?
If you could snap a zoom there. That is a striped tail.
Cool.
And they have those vertical bars, and they have dark pigmentation in their caudal fin, in the fin end. Right? You can see it really well on the side.
Ouch. Just got poked by an urchin.
Hmm.
That was
Cool.
surreal. I was
Good.
seeing all the squid moving, and I thought Hercules was going lateraling left.
You can get a tight zoom there if you want.
Sure, let's go for it. It's being too accommodating. Yeah, that's great. Thank you, guys.
Okay.
Does not appear to be bothered by the large yellow robot.
Hiding from all those squid above us.
So I think we're starting to see some combfish now, probably short spine combfish, which look a lot like a poacher. But whereas poachers will be very straight, the combfish will actually curve into a letter C.
That is extremely accommodating. Is that a green stripe that I saw on
Yes.
the left?
What gave that one away?
I didn't want to let you down. I went for the easy pickings.
Yep. Lots of combfish in this area. Another Stylostereus sea star.
How many hours of video do you think you've had to watch, Erica, to get to the level of knowledge you have now?
Well, the AUV only takes still images, and we come back from a cruise with about 50,000 images
Whew.
per 7 to 10 day cruise.
Yeah.
So there's the squid. But I have to say that I learned the bulk of my IDs when I did the West Coast Groundfish Trawl Survey for 10 years.
Oh, okay.
In my
Yeah.
first, or as my first job with NOAA. So that's where I learned the bulk of the species, and then having to translate that from having a fish in your hand to trying to identify it in images has been challenging.
Yeah, I'll bet.
And our AUV images were looking straight down onto the seafloor, so imagine having to identify a rockfish from a top-down view. There's actually one stuck under that rock, or hiding under that rock.
Quick zoom there. Can't see me, my head's under the rock.
What are you? That might be a boccaccio, or it's something definitely bigger than we've seen. Cowcod, yeah, that was my second guess. Erica Burton's chiming in.
Almost looks like a den.
Okay,
Thank you.
pleasure. This is a lot of squid in the Argus camera there.
Oh, look, they're just out of the light, and so now you're approaching them. They're like, oh, not too much light.
One tried for the laser.
Squid rain.
See him attacking the lasers?
Oh my God, look at them all around Herc on there. It's great.
It's funny.
Chris, do you know anything about the commercial squid fishery down in your area?
Just a couple things. If it is booming here, so it moves up and down the coast where all the squid are going to be. And if it's booming here, maybe it's not somewhere else. So the fishermen will actually come from far away. We might get fishermen coming up all the way from Monterey or north. Will come down if this is the hot spot at the time. And when it's the hot spot, it'll be the most valuable fishery by dollars that we have in this region, state fishery that we have in this region. And you'll get the squid boats will come out and mark their spots around the islands, and harvest at night.
Quick zoom there.
Ship's held up. You want me to continue?
They'll have a light boat that goes out with really bright lights and attract the fish, and then the runners will go out and encircle the squid with a net and pull them in. We've been listening. Sometimes they use that when they're out fishing, the seals or sea lions will be attracted to the lights as well into all the food. So the seals and sea lions will come out off the islands to grab the squid as well. But there's concern about them getting caught in the nets or stealing all the catch, and so they'll use these little noise makers, little explosives that they'll throw in the water to scare the sea lions away. And we have acoustic listening devices now around the islands that pick up all kinds of noises, but also pick up what we call them seal bombs. We'll pick up the noise from the seal bombs, and will give us an indication of how much fishing activity is going on. And at least last year we didn't pick up too much. I don't know if we've analyzed the data yet for this year from the region. The squid will come in, and even in shallow water will lay egg cases. And as scuba divers, we'll go down and see they kind of look like long, maybe 8-inch long fingers along the bottom in clusters. And so we know, and we've seen them every year. I've gone out and every year we've dove. We definitely see that they're laying egg cases, but just some years not in the same abundance as others.
Yeah, that was a great zoom on a benthic siphonophore. And I think all your boats were up here in Oregon last year. I've lived here for almost 20 years, and there's not been a commercial squid fishery until the last couple of years.
Wow, I did not know that they had made it all the way up there.
Right. Yeah, we were seeing seiners in our harbor that we've never seen before.
Indications of a changing climate.
Oops, sorry, wrong button.
Starting to see
Are you
the base
based
of
in
this
Newport
slope.
then, Erica?
I'm sorry.
Erica,
Could you say that again?
Are you based in Are you based in Newport then?
I am, yes.
Okay.
So this is different, more cobbly, couple of boulders, some dead header cone. I saw some aria at the bottom.
What are all the white things growing on the rocks?
Could we take a look,
I don't
actually? Could we
know.
zoom in on?
A zoom would be great.
Are they limpets?
They could be brachiopods.
Yeah, go full zoom there, video. Flatfish hiding there.
Daniel
have to
saying,
say,
yes,
I
please
do.
zoom.
I don't know what I see. One limpet, but the other ones I don't know what those are. Shingle shells.
Daniel, any reason to collect? We could probably pick up a small rock if desired. No collection. Okay.
And so that's also one of the smaller Dover soles we were talking about earlier.
Really blends in.
They definitely are segregated by depth and move deeper and farther offshore as they get larger.
You got time. You got time. No, I don't mind.
Dan, can we get a zoom on one of these white spotted rockfish, please?
Sorry, zoom on what?
One of the white spotted rockfish, upper right-hand side, maybe?
Ah, I see a white spotted rockfish. Your video, you can zoom there.
Small black coral.
Yeah, that one's the
Okay, I'm gonna hand you guys back over to
package.
Jess. Thank you very much. That was fun.
Thanks. So I'm going to lump this one into that Sebastomus category,
All right.
but the smaller ones we see down here are sword spines, rosies, but they usually have a pink or purple blotched head area. I don't know, maybe somebody more versed in Southern California rockfish can chime in here.
Oh, the squid are really amassing in Argus.
Oh, yeah, there they are.
All in one direction.
All right, so we're continuing upslope here?
Yeah, we're going to continue upslope for another, let's do 30 meters to see that.
Sure.
And then, yeah, and then we'll start heading along this cliff to the west of us.
Sure.
So I'll back down. And
yeah, sure. So they want to do another 30 meters up and then...
yeah, I'm just trying to see the contours, 10, 20. Yeah, maybe we should just get to that target, 40 meters up.
Okay, then they want to go kind of
along this contour. So we'll just...
Roger.
Yeah. So I can...
Roger that.
All right, I'm going to bring us, let's see. Yeah, I'll do 50, or, yeah, I'll do 50 meters, 010. Raj?
Roger that.
Erica Burton suggesting sword spine off also.
All right, Argus is moving.
Sorry?
It looks like Argus is starting to move.
Roger that. Looks like all these rocks have polka dots on them.
We did a Zoom while you guys were at dinner and were informed that they are shingle shells.
Shingle shells. Not a single sound.
Right. So Marley needs to write a poem about shingle shells.
Be hard for someone with a lisp.
I'm on it. What are they?
A gastropod?
Some sort of mollusk, I believe. Clams. And Daniel's saying they're sessile, a bit like oysters. And that's what first came to my mind when we did the Zoom on them, but I knew that couldn't be right.
They definitely look sessile to me.
It'd be so great to have the backscatter here and see how that changed for the rubble.
Went from medium gray to light gray. No. I know that stuff can be such an art to interpret.
So these little white spotted rockfish really blend in with the white spotted rocks.
Huh.
You don't see them until they move.
Seems useful. So many of these.
Several different encrusting sponges. Another siphon 4. I think that was some plumarella off to the left-hand side.
Suggest we just have 10 meters left in that move.
Roger.
I think we'll be in a good spot to start heading towards the slope.
Sorry, but I don't think I hear you very well.
Yeah, I think we can start heading towards the slope after this move. So.
Roger that.
Another piece of dead heterocone. And we keep calling that particular sponge heterocone, but we sampled one earlier in the dive. We're thinking that there may be 2 different species down here. So our sponge expert, Abby Powell, is going to try to determine if they are all heterocone or if there's something else in play. And we have to learn to differentiate between 2 very similar looking sponges. Lots of crinoids on these rocks, on the bigger boulders mostly.
Marley, you want to do a snap zoom there, please?
Roger.
That's great. All right, Marley, go ahead and come wide.
we're ready, we can do just 20 meters east.
Roger, so we're gonna go along that steeper contour now.
Yep. And we'll start backing down to the south.
The geology here is so cool. It's so different.
It's quite a mix. You've got several
Yeah,
different sizes here.
Roger. So what's the
See.
first move gonna be?
Yeah, I'm just going to take us 20 meters east.
Roger.
Yeah, I just want you to be ahead of me. Then we'll be good.
Sure thing. I'll get over in position.
Yeah, I'll wait.
All right, Ben. Go ahead.
I'll probably bring us like 140 after that.
Roger.
So is that more of a shallow water rockfish, that brown one?
All of these are more
Yeah.
shallow species. We're seeing what we think are sword spines. We've seen some green stripes. The barred one heading off to the left, I think, is a striped tail. And the shallower we move, the more species we're going to come into.
Hey, Ben, do you have any idea, like, roughly how far we've traveled since the start of the dive this morning?
I can do a quick crow's flight, but yeah, we've done a lot of coverage. Just don't wanna,
I was just curious since, I mean, I know we're much shallower than we were this morning, but
yeah,
it's just so
well, we've
different,
come down
you know?
this. So yeah, we've come about 3 kilometers crow's flight, probably like more like 4 total.
Huh. Nice.
Yeah. Yeah, we started on this kind of the forward slope of this reef behind us, traversed this valley to this upslope, ridgy part, and I guess we're below the footprint reef.
We're starting to see a little bit larger rockfish coming in from the right-hand side with the bands. The dark edge tail. Oh, I saw Di was here and now she's gone. Looks like a plate sponge covered in crinoids right in front of us.
So right now we're moving along a bathymetric contour just below the south side of footprint. We'll be continuing on along this contour for a little ways. The slope's a little bit steeper here. We've run into some more rubble
Bridge nav,
earlier
you
with
can
the
add
dive.
20, or let's do 30 meters, 150.
Earlier on the
Or
dive,
actually
we were
140.
just below
Oops.
this point. A lot more soft bottom and sediment, and fewer rocks and rubble. Among other things, we're trying to get a sense for the distribution and abundance of the resources around the Channel Islands National Marine Sanctuary. Critical to resource management and protection to have a basic understanding of what and where all your plants and animals in the zoo are. And so by getting eyes underwater like this with the ROV, we're able to do that in both a characterizing way, so going out and just exploring and noting what we see, and also a quantitative way. So in addition to just traversing along, we're doing quantitative transects over certain distances where we'll count and identify everything that we can to understand is one place, do we see certain animals in one place more abundant than another, so they can make better informed decisions. Channel Islands were largely recognized early when they were first protected for the shallow waters, the kelp forest communities. So people came to dive in for recreational fishing opportunities and so forth. Those waters go down to, kelp tends to go down to maybe 30 or 40 meters deep, at least the giant kelp macrocystis porifera, that people like to check out and dive in and have the charismatic fish fauna like the Garibaldi and sheephead down here, the lobster. But 90% of the sanctuary is actually in waters too deep to dive. And so for that, we need other technologies to get down and see some of these species, the rockfish, the crinoids that you're seeing, deep water octopus, deep sea coral, sponges, and so forth.
Could we zoom on that rockfish right in front of the lasers, the bigger one?